Hello, and welcome to the third lecture on viruses. In this lecture, we will cover bacteriophages, or more generally, viruses that can infect bacteria. So, bacteriophages only replicate in the cytoplasm since prokaryotes don't have a nucleus or organelles. So, when they are made, they're made readily and they begin to go under their cycles, and typically they will become a virulent phage, where the phage actually uh, dies or makes the host die at the end of the cycle. There's also temperate phages, which are more likely to become integrated into the host's genome than they are to be deadly. And with that in mind, bacteriophages multiply in two different ways, either in the lytic or lysogenic cycle. These cycles are kind of related to each other. In fact, the lysogenic cycle is more of a spur of the lytic cycle. And the lytic cycle is kind of typically what you would expect from a virus infecting a cell. So when we look at the lytic cycle, we can read it on the far side and look down, or we can read it right here and go across. All right, so if you're visual, you will look at the pictures first and kind of follow where that blue DNA goes, okay? And the blue DNA is viral, and at this point, the pink in these last three steps, bacterial DNA becomes irrelevant, and so uh, that kind of tells you where the viral DNA went, and also can kind of tell you how we can kind of take advantage of that later on, how we could test where that genetic data is coming from. Is the genetic data from viruses or is it from bacteria? And we'd be able to see readily using modern technology. Anyway, the lytic cycle has 5 steps. Okay, the first two, attachment and penetration, being the main two for prokaryotic viruses and for eukaryotic viruses. Okay, so attachment is key. Okay, and what do we use to attach with? We use our spikes. Okay, we need the spikes to attach. We need to attach to infect if we are a virus. Okay, now that we have attached, attachment is connecting to key receptors on its host. Okay, remember, it can only target specific hosts, and that's why those receptors on the outside are very specific to the virus. The virus now is able to attach with its spikes, and then it will penetrate, and that just means that the DNA will be able to go through any membrane and any cell wall and into the cytoplasm. Okay. Penetrates into there, um, and right away the ribosomes inside of the cell begin to make protein and any other bits and pieces that the virus brings in with it on its genome. Okay, and it can either be RNA or DNA. Then they start to mature. All of our pieces are there. We have our concrete, and we have our roof tiles, and we have our pipes and electrical cord, right? We have everything that we need to make a new virus. And so now we need to put it all together. We're going to mature. Okay. Mature, the assembly phase, you could call it, starts. Finally, there's going to be too many viruses inside the cell, and they actually just burst out in all directions. Okay, and that bursting is called lysis. And that is our lytic cycle. You will recognize the first two steps, attachment and penetration. Okay, and then that is when it becomes a little different. it will end up entering the lytic cycle, but for now it is going to become latent. Okay, it won't be available to be expressed to start making these new viruses. Okay, so it infects a cell, and then the DNA is now in the cell, but no viruses are being made. So when the cell divides, it's now replicated DNA. That replicated DNA is now has the viral DNA also, and so now two daughter cells both have it. It's latent. Okay, this is also known as the prophage. If you follow the blue is the prophage. Okay, and then even here it says under stressful condition, the prophage, which is the blue equals prophage. Okay, the prophage will then go under lysogenic conversion and induce or induction this prophage to come out. The prophage comes out. The phage now does re-enter the lytic cycle, and eventually the outcome is the same. This can take years, it can take months. Who's to say how long it takes? In bacteria, it's probably really quick, but because they don't live very long. But it's very years and months when it becomes latent in a eukaryote. Okay. One more thing before we move on to eukaryote, more specifically animal hosts, we have transduction. Okay, transduction is a special way that new cells get new DNA. We've learned about the sex pilus and transferring DNA between bacteria using the pilus, but this is another way to get new DNA. It's called transduction. You can have it either generalized or specialized, but the key is that when it gets excised as a prophage, it includes some of the old host DNA. So when it finally gets back into a new host, the new host has both the viral DNA and a tad bit of the old host DNA. And that is how also viruses can replicate. Alright, now we'll move on to animal hosts with viruses. I keep telling you that it is pretty much the same two first steps in the beginning, attachment and penetration. Okay, we need to get on a cell and stay on there, and then we need to get inside a cell. Okay, and the cell needs to be our host. Right? So once the cell gets in a host, its viral contents get released, and now we kind of enter the same set of steps. They just call it differently: biosynthesis, maturation, and then release. Okay, instead of lysis. And it's not called lysis because the cell is too big for the diseases typically, so it doesn't lyse them. In animal hosts, we have a distinction of if you have a single-stranded RNA for a genome, that single-stranded RNA can either be ready, okay, can be translated directly, can be ready to go. As soon as it gets in, that would be positive-stranded RNA. But if you are a negative-stranded RNA, you don't have the machinery to directly translate the genome yet. So you have to wait until the polymerase is brought in, and that is how you can make DNA from a negative strand, single-stranded RNA. Alright, and we also want to talk about retroviruses in our virus lecture. Retroviruses are called so because they go against a central dogma. What's the central dogma? The central dogma is that you have nucleic acid that is the DNA. That DNA makes a transcript or a workable set of blueprints. That workable set of blueprints goes out to the machinery and the ribosome specifically and makes a protein. That is the central dogma. Why a retrovirus goes against it is because instead of DNA to RNA to protein, you come in with a RNA already. Okay, so but we can't just go to protein. We first need to go to DNA, and we call that cDNA. Then we can come to RNA again, and finally a protein. Okay, so because it goes against the grain, upriver, so to speak, that makes it called a retrovirus. When a retrovirus integrates into a genome and becomes latent, it is a provirus, not a prophage. Remember, phage just means bacteria, so the provirus and prophage are different. The provirus is a retrovirus that is integrated into DNA. And they can stay in there for a long, long, long time and even establish that chronic infection we were talking about. And with our animal hosts, we can see the steps just like the others. We need to attach, right? And then penetrate, enter, penetrate. Then the reverse transcription happens. The viral DNA is transported across into the nucleus, integrates into the host DNA as a provirus, which is highlighted here. Okay, that provirus makes new RNA, and that RNA makes more of itself and more virus proteins. So that's how a retrovirus works. And we will end the lecture on this slide with viral infections. The viral infections that we can have, that we'll learn about down the road, can either be persistent, which is HIV, herpes simplex, which are good examples, and they can either be latent or chronic. Okay, so while they're both persistent, chronic means that they're kind of recurrent, whereas latent means it's a dormant disease, and herpes can come back at any time. Chickenpox can come back as shingles later on, whereas HIV, you're kind of like suffering through it chronically, like in hepatitis you would be as well. Alright, so I'm going to end the video here because it's getting a bit lengthy, and we will pick up with the short video to end the viral lectures.